The first thing your body has going for it is its skin, and then you have mucus lining some of the more vulnerable membranes. So here you have lysozymes in tears. So you have all that, around your eye. That would be a great place for bacteria to come in, but you have lysozyme to help kill bacteria that might be there. And skin surface is a physical barrier. And you also have fatty acids that help to limit bacterial growth. And then normal, this is fauna. And that's a fancy word for bacteria and any other living things that you might have on your skin. I'm sorry, not fauna, that'd be animal, but flora. So things like bacteria, fungi. These are normal. We're not talking about abnormal things growing on your skin. And you'll notice right here, like from our activity today, we did the human digestion. When food goes from the stomach into your small intestine, there's a huge switch in pH from 2 to almost 8 or a little bit above. So boom, that's a big switch. Not many living things can handle that kind of a change. And here we have the stomach, pH of 2. And back here, like the urinary tract, I'm going to have to go back and edit that. I can't remember what's behind there. But you have your urethra in males and females, and you have a low pH, slightly acidic, and that helps to inhibit bacterial growth. Urine is a little bit acidic, helps to inhibit bacterial growth. And you have mucus in your trachea. That helps to limit the growth of bacteria, helps to stick to bacteria or anything else that might be in there. And then you have the nasopharynx, and in your trachea you have cilia that helps to sweep out bad stuff. So sweeps. That's a terrible. Oh my gosh! Can't you sweep? So it sweeps out bacteria. Anything stuck in that mucus, you breathe in. This is pretty amazing. You can see here red blood cells. I'll write it out here so you can see it. Red blood cells. It's not apostrophe. Just that's red blood cells plural. And you have all this fibrin coating it like a net, and this is keeping the red blood cells from escaping. Help to keep you from bleeding too heavily if you get cut. That's just a cool electron scan. It's like a scanning electron microscopic image. See all these cool disc-shaped red blood cells. And you have these things called platelets. They normally look like this, and then when they get activated by clotting factors, they kind of grow these funny little attachments that go out and grab onto other platelets and then can form a net along with the fibrin. So you have a pretty major response whenever you get cut. And this is a diagram that kind of describes the two main types of activation. It gives you a lot more detail than you need. This is what normally happens. So this is normal stuff that you've all seen, and this is from any kind of trauma. You get cut, somebody need a Band-Aid. You have all these factors creating different pathways, and the thing to remember, the main thing you need to know, you got prothrombin changing to thrombin, and then fibrinogen changing to fibrin, and that's what the clot is made out of, this fibrin right here. But you see all these complex things, and this is called contact activation, and this would be some damage to the surface inside your blood vessels. So I covered up damage to surface. This would be not your normal way to go here. It's kind of a, don't really know the exact cause, but this can be misfolded proteins, bacteria swimming around in your blood. This causes a whole nother activation pathway that leads to the same thing. And as you know, this is not good. You don't want clots floating around in your blood. And you do want this over here. Check. You want this if you get cut. You want that blood to stop flowing. But this can cause things like strokes, and it's a little bit unknown. You can follow this link to a really interesting article. So you have, if you have some kind of damage, this would be that extrinsic pathway. You have clotting factors here. Let's see, get my handwriting up to speed. Yeah, there we go. Clotting factors, okay, and that's helped out. It's in a conjunction with platelets, and that takes prothrombin, turns into thrombin, and thrombin in turn takes fibrinogen. Let me rewrite that big so you can see it. Fibrinogen, and turns it into fibrin. And this is a solid. It's not soluble. And all these other things, your thrombin, fibrinogen, prothrombin, those are soluble. Those are liquid. So they're not going to cause a clot. But finally, this fibrin will wrap up with the blood cells and the platelets to, I think I saw, is that a rat or a squirrel? I don't know. To form this plug. You don't want your blood coming out. You want it plugged up. So that's what forms. Oh crap, those are mice. Okay. Good to know. Along my fence. Okay. Stay out of the house. All right, they're mice. Not very big. So we get that stick. Okay, so we got our pathogen right here. This looks like bacteria. So it comes in here, and you'll see that it'll form a vacuole right there. And this vacuole will form with a lysosome. So the lysosome will put these enzymes inside like you see here, and it'll break it up. And all this is debris. This is old stuff, harmless, and it'll break out. So these phagocytes kind of eat bacteria, digest them, and take care of business. This is something called cell-mediated immune response. It's a little more complicated, takes a little bit longer. But basically, you have a macrophage swallow this infected cell, and then it'll create all these other cells that help to target things more specifically. This would be a memory T cell, and this is a helper T. And you'll see once it sees the need to multiply, it'll go through mitosis, and we'll go into more detail with that later. But it'll find this infected cell, specific to that antigen, and this would be a killer T. And it'll use these chemicals called cytokines to come in here and then develop and destroy it. And here, this would be a dead cell. Doesn't look too good, does it? So these are targeted responses to cells that have been infected and that need to get removed from your body. Here's another way to look at it. This kind of separates it into humoral, which is antibody-based, and the cell-mediated, which I just showed you. This is the phagocytes here, and basically T cells over here. But you have the antigen, and that basically activates these B cells, Helper T cell helps both pathways. This is a cytotoxic T cell. Oh, I'm sorry. Yeah, helper T cell, and this is a memory cytotoxic T cell. Plasma cells are what make the antibodies for you. So you all heard of antibodies. They're these kind of like little Y-shaped looking things. They see something bad in your body, they come and like will surround it and kind of label it for destruction. So it's B cells. So we got the antigen, leads to the B cell, leads to the plasma cell, which leads to the antibody. And after everything's done, you have these memory cells. So you get if you get infected again with that same pathogen, you're all ready to go. Your body is primed. So we have external defenses, things like your skin, mucous membrane, secretion, like we talked about before. Internal, if something gets inside, phagocytic cells, natural killer, and antibodies would be your humoral response, and then cell-mediated response to those cytotoxic T cells. And antibiotics is important. They block activities that occur just in bacteria like prokaryotic cells, but not cells like ours. So metabolic enzymes that are specific, sulfa drugs, which you probably haven't had, but you may have had penicillins. They go after the cell wall structure, and tetracyclines are very common. They go after ribosomes, and you'll notice 70S because eukaryotes have 80S, so they won't hurt our ribosomes. And then, you can see this. What structure is this on the very inside? You guessed it, the membrane. So cell membranes, polymyxins, I'm not too familiar with that. And then DNA synthesis. I think it's quinones, DNA synthesis. So it blocks bacteria from making new DNA. It can't make DNA. It can't make new bacteria. So blood clot formation. This is what we're talking about in John's heart attack. And this gave a nice image of this. You have this plaque form, and there's always a membrane surrounding that plaque. Plaque with fibrous cap. That's a BR. That's a FIBROUS. And then the cap is ruptured. It's damaged. It was John. For the John, it was during the soccer game when he was playing really hard, a lot of blood going through. And then you'll see the blood clot right here. Ooh, blood clot form, blocking the artery. And that'll lead to cell death. So, if it happens too fast, that can cause death. We saw, which I thought was fantastic, it mentioned in that article and that video. If it's slow enough, the body can make new arteries, make new blood vessels. Okay, I can't remember the word, but something genesis. It'll create new vessels. It goes around. It basically, it can make a natural bypass. Surgeons can go in and then attach a blood vessel that goes around, blocked artery, but your body can do it by itself. It's awesome. So here's some experiments where they knew about penicillin, but you got to show if it really works. So these mice were infected with a pathogenic, which means disease-causing or deadly bacteria, hemolytic streptococcus. So it basically destroys your blood vessels. So the control was they just injected these poor mice, and basically they all died. I don't know why they didn't do, they just did 4, not more. Anyway, these experimental group got the bacterial injections, okay, and the results, all 4 survived. So that was pretty good evidence that this penicillin worked. They infected these mice in the same way. The ones that got the treatment, the penicillin, survived. The ones that didn't, died. And they actually tried this on a person. That they look over, not at this hospital. One person is almost dying, was close to death from a bacterial infection they got when getting scratched by a thorn, a rose thorn, which is kind of seems silly, but it happens. And they gave this guy penicillin, and he got a lot better. But unfortunately, they ran out and didn't have enough to kill off everything, and then it came back, and he died from it. So it has antibiotic properties, kills bacteria, but not the person or the organism. Okay, so when you talk about HIV, how do you get it? This looks like they're getting a little intimate, so sexual contact is one way. Pregnancy, so you can get it during pregnancy. The fetus can, that is, but it's very rare, or during childbirth, or during breastfeeding is more common. And during childbirth, if you haven't tested the mother. If the mother's been tested, they'll do a C-section. And if they do that, the baby won't be born with HIV. It's been very effective. Here is IV drug use. You see with a needle. It's one of the main ways it's passed. So here's occupational exposure, also very low. You're an occupation like a nurse, a doctor, where you're around blood. Needle sticks are a common problem. So, and then here's transfusions, which is also very uncommon. They test blood, and it's very uncommon for somebody to get HIV from a blood transfusion. So if you get HIV, it goes after your T cells, and basically you lose your CD4 cells, and that makes you prone to infection, to normal infections that turn deadly. So people will die of pneumonia that normally no healthy person will get, or a certain skin cancer called Kaposi sarcoma, which are all telltale signs. Here we got some pictures of HIV budding from a CD4 cell. So that's HIV virus. We see produced there. And I added these just HIV/AIDS has such a political angle because of the people assign morality to getting this disease, because it first occurred mostly in gay men, but we know it'll affect anybody. This Ryan White here, he got HIV. He's a hemophiliac, and they need a lot of they need a lot of blood products, and back in these days they didn't test them, and they gave almost every hemophiliac that had HIV got infected. Every hemophiliac that got treated was infected with HIV, and many of them died of AIDS. And Ryan White was the first big case where it was somebody dying of HIV that wasn't infected with IV drug use or from homosexual sexual activity. There were people, especially in the Republican Party, which Ronald Reagan was a part of, that would say, oh, it's a moral thing. If you get it, it means you're a bad person and you deserve to die. They actually said that. People in Reagan's party said that. and he lacked the courage to do anything about it. So Ronald Reagan lacked courage, and what also he did, he knew, and others, that you don't get HIV from shaking hands, from being in the same room with somebody, and he refused to make a statement. So a lot of people were freaked out about HIV. There's a real lack of leadership from Reagan. And you see this picture here. This comes from a couple brothers, 3 in fact, that had hemophilia, and they all got HIV/AIDS. And people, since the political leaders like Reagan and others were kind of scaring people in a way, people kind of go out of their minds, and somebody burned their house down. So not only did they have HIV/AIDS, somebody went and burned their house down. Luckily, this is Liz Taylor. You had some people that provided real leadership in protecting people and informing the public and making sure people knew this is a disease we need to fight, and we can't just stick our heads in the sand and make these moral arguments, which are ridiculous. We never say that about any other disease, but they did that about HIV. So Liz Taylor courageously went against Ronald Reagan and a lot of people in this country, and said we need to care about these are human beings. We need treatment. We need education. We need it now. And she's something, a real hero to getting the country to realize it's a problem and do something about it.